Hi guys, my name is Vaishnavi. In this video, I'll be talking about histological structure of the posterior part of the eye. The posterior part of the eye begins behind the lens and extends to the optic nerve at the back of the eye. The vitreous body occupies the vitreous chamber behind the lens. It is composed of connective tissue containing a jelly-like amorphous transparent substance that is rich in hyaluronic acid and small amounts of collagen and few cells. Now coming to sclera. Sclera protects the internal delicate structures and provides a site of extraocular muscle insertion. The majority, 5 out of 6 of the fibrous layer, consist of the sclera. It is relatively avascular, tough, dense connective tissue with collagen type 1 bundles and scattered fibroblasts. The fibers are parallel with the surface. Posteriorly, it joins the epineurium, covering the optic nerve. At the circumference of the cornea is the limbus, the corneoscleral junction, where the transparent corneal stroma merges with the opaque sealed sclera. The epithelium of the limbus is slightly thicker than corneal epithelium, containing stem cells for the cornea and is continuous with the conjunctiva covering the anterior part of the sclera and lining the eyelids. The stroma of the limbus contains the scleral venous sinus or canal of Schlem, which receives aqueous humor from an adjacent trabecular meshwork at the surface of the anterior chamber. Choroid is a highly vascular tunic in the posterior two-thirds of the eye with loose vascularized connective tissue rich in collagen, elastic fibers, fibroblast, melanocytes, lymphocytes, mast cells, and plasma cells. The abundant melanocytes gives the layer its characteristic black color, especially in the suprachoroidal lamina, and blocks light from entering the eye except through the pupil. Suprachoroidal lamina is the outer part bound to the sclera. The choriocapillary lamina is the inner vascular rich region. The choriocapillary lamina is separated from the retina by a thin membrane called the Bruch's membrane, which extends from the ora serrata back to the optic nerve. Retina is the inner layer of the eye and consists of two major layers. The inner neural retina, which contains the neurons and photoreceptors. The visual region extends anterior only as far as the ora serrata, but it is continuous as cuboidal epithelium lining the surface of the ciliary body and posterior iris. The outer pigmented layer uh, has epithelium resting on Bruch's membrane inside the choroid. This pigmented cuboidal epithelium also lines the ciliary body and posterior iris, contributing to the double epithelium of those structures. The cell of the retinal pigmental epithelium has diverse functions. So, it is important part of the blood retina barrier, absorbs light passing through the retina to prevent its reflection. Phagocytoses shed components from the rods and cones, remove free radicals. The inner neural retina is the photosensitive part and has three major layers of neurons. Outer layer of photosensitive cells, so rod and cone cells. The intermediate layer of bipolar neurons, which connect to the rod and cone cells. The internal layer of ganglion cells, which synapse with the bipolar cells through their dendrites and send out axons that converge to form the optic nerve. Between the rod and cone cell layer and the bipolar cells is the region called the outer plexiform layer that contain fibers and the synapses connecting the neurons in these two cellular layers. There's a similar region of synapses between the bipolar and ganglion cells, which is called the inner plexiform layer. The retina has an inverted structure with the light first passing through the ganglion layer and then the bipolar layer to reach the rod and cone cells. The rod and cone cells can be divided into outer and inner segments, a nuclear region and a synaptic region. The outer segments are modified primary cilia and contain stacks of membranous sacules. The photosensitive pigments of the retina are located in the membranes of these sacules. Both cells pass through a thin layer, the outer limiting layer, which consists of a series of junctional complexes between the photoreceptors and the organizing glial cells of the retina called the Mueller cells. Mueller cells are type of retinal glial cells serves as support for the neurons of the retina. They span across the entire thickness of the neural retina. Their major function is to maintain stability of the retinal extracellular environment by regulation of the potassium levels, uptake of neurotransmitters, removal of debris, storage of glycogen, electrical insulation of receptors and other neurons, and mechanical support of the neural retina. The rod cells are extremely sensitive to light and allow some vision even with low light levels, so nighttime vision. They are thin, elongated cells composed of two distinct segments. Outer segment is photosensitive, consists mainly of flattened membranous disc. Between the outer and inner segment, there's a construction called the connecting stalk. The inner segment contains the nucleus, a metabolic machinery for the cell, rich in glycogen and mitochondria. Polyribosomes located inside the mitochondrial region produce proteins that are transported to the outer segment. These proteins include the visual pigment rhodopsin, which is bleached by light and initiates the visual stimulus. The cone cells are less sensitive to low light than rod cells. They are specialized for color vision in bright light. Three functional types of cone cells, not distinguishable morphologically, contain variations of the visual pigment iodopsin with maximal sensitivities in the red, blue, and green regions of the visible spectrum. Cone cells are also arranged with outer and inner segments and connecting stalk. The outer segments of cones differ from those of the rods in their shorter conical form and the structure of their stacked membranous discs, which in cones remain as continuous invaginations of the extracellular space. Phototransduction involves a cascade of changes in the cells triggered when light hits and activates retinal. It is basically similar in both rod and cone cells. Neurons and glial cells in the retina. So, the inner nuclear layer of bipolar cells consists mainly of various bipolar neurons, which have processes extending into the inner and outer plexiform layers, forming synaptic connections with the neurons in all layers of the retina. Horizontal cells and the amacrine cells also they have their nuclei in the inner nuclear layer, both with processes that mainly spread horizontally in the plexiform layers and integrate signals from photoreceptors over a wide area of the retina. The ganglion cells is the innermost ganglionic layer and are typical nerve cells, synapse with the bipolar and amacrine cells and project their axons to the nerve fiber layer where they come together to form the optic nerve. They are kind of photoreceptors themselves, involving not with vision but in detecting the changes in the light quantity and quality during 24-hour cycle. They signal the hypothalamus nuclei, establishing the body's physiological circadian rhythms. The posterior area of the retina, where the optic nerve leaves the retina, is devoid of photoreceptors and is known as the blind spot of the retina or the optic disc. There's a specialized area of the retina called the fovea centralis, a shallow depression having only cone cells at its center, with the bipolar and ganglion cells located only at the periphery. Surrounding the fovea centralis is the macula lutea, which has all layers of the retina, and the two plexiform layers are rich in carotenoids, which give this area its yellowish color. These carotenoids filter potentially damaging short wavelength light. Thank you.